Hello, my name is Gladys Alexandri. I am the professor and the head of the BCMB department here at UTK. I welcome you to this virtual visit of our department that showcases several of our researchers. Research in BCMB addresses fundamental questions in biology, spanning scales from the atomic cellular to the organism, in order to seek solutions to challenges in medicine, engineering, health, the environment, and agriculture. BCMB is a collegial, supportive, and collaborative environment. I invite you to contact us to learn more about how to apply to our graduate program. My own research focuses on strategies that cells use to adapt to their environment and the molecular mechanisms by which these cells make decisions, which can include growth, motility, or differentiation. I am a microbial cell biologist, and thus I use bacteria as model systems. My laboratory is using microbial genetics, biochemistry, and cellular biology to address the following general questions. How do motile bacteria integrate signals from multiple transduction pathways to produce a coordinated response? What are the signals exchanged between plants and microbes to produce beneficial associations? And last but not least, how does bacterial membrane fluidity regulate cell physiology, including antibiotic resistance? I invite you to discover other exciting research area and opportunity in the following sequences. Once again, welcome to BCMB.
Hello, my name is Andreas Nebenführ, and my lab is studying how plant cells make sure all their parts are in the right places. This may involve large-scale mixing or slow rearrangement of the furniture inside a cell. Of course, for this, we do a lot of live cell microscopy to keep an eye on events as they are happening, such as in this video. What you're seeing here is 3 different organelles with red Golgi stacks, green mitochondria, and blue peroxisome moving through a plant cell at right high speed. And when you see these movements, you can clearly see that why people call this cytoplasmic streaming. My lab is trying to figure out how all this works and what mechanisms are in place to regulate these large-scale movements. We already know that myosin motors can attach to organelles and drag them through the cell. Now we're studying more specifically what the individual myosin motors in plant cells are doing by looking at mutants. We also try to understand how this attachment to the organelles functions. And of course, we're also trying to manipulate these movements with targeted genetic engineering interventions to ensure that we get a full understanding of how the plant regulates these processes.
Hi, I'm Rachel Patton-McCord, and my lab studies how 6 feet of human DNA can fold up into a tiny microscopic nucleus, and how this genome structure, and not only the DNA sequence, can go wrong in cancer, or after DNA gets damaged by radiation, or in premature aging diseases. We use many different tools, microscopy to watch crawling metastatic cancer cells, lots of DNA sequencing and molecular biology, and computational tools to analyze large amounts of data.
Hi there, I'm Jeremy Smith, Director of the Centre for Molecular Biophysics and Governor's Chair at UTK. Centre for Molecular Biophysics has about 20 people in it, and it's located at Oak Ridge National Lab. We do computations. We require physics, chemistry, biology background. And we do research into drug discovery, for example, against the coronavirus, but also bioenergy and environmental science, circulation of mercury. And we are heavy users of the Oak Ridge supercomputer, which is currently the nation's fastest one.
I am Dr. Kirti Krishnan, a neurobiologist interested in understanding brain plasticity. This mechanism is fundamental to how our brains function in an ever-changing dynamic environment. When this process goes wrong, our brains exhibit abnormal plasticity, resulting in atypical behaviors and neurological disorders. We use techniques that connect the dots between brain cells and behavior, such as fluorescent microscopy, electrophysiology, and deep learning algorithms to decipher animal behaviors.
Hi, my name is Razal Lamisani. I'm an assistant professor. My field of research is in biochemistry and biophysics. In my lab, we use state-of-the-art single molecule techniques to address important biological questions regarding protein folding, protein functions, protein DNA, and protein RNA interactions at the molecular level. This means we are able to observe biomolecular activities one molecule at a time. Currently, we are working with understanding the dynamics of integral membrane proteins called G protein-coupled receptors, which are the target sites for treating many diseases, including cancer, metabolic diseases, and several inflammatory and neurological disorders.
I'm Tessa Birch Smith, and I'm an associate professor in BCMB. I am one of several faculty in the department who study questions of cell and molecular biology, biochemistry, and genetics using plants as model organisms. In my lab, we study intercellular trafficking in plants and how viruses usurp plant systems to cause disease. We use a variety of tools for our research, including state-of-the-art light and electron microscopes located right here on campus.
My name is Brad Binder, and my lab studies signal transduction mechanisms for the gas ethylene. Ethylene is well known to affect plants, and the receptors there are well studied, but we've also expanded our research to look at newly discovered receptors in bacteria. In both cases, we're trying to understand how a gas binds to a protein and then how this signals to downstream proteins to cause changes in the organism. So if you're interested in finding out more about my research, please visit my website or contact me, and I'd be happy to set up a time to talk with you.
My name is Tian Hong. I'm a computational biologist. In this department, we have several research groups that are focused on using computer models to solve biological problems, ranging from structure of molecules in our cells to how our body works. In our research group, we're interested in how the networks of our genes control the decision-making of our cells. For example, how our immune cells are differentiated so that they can fight against pathogens.
Hello, I'm Maitreyi Das, an associate professor in BCMB. I'm a cell biologist, and my research focuses on understanding the basic processes of cell shape establishment and cytokinesis. These processes are fundamental to all biological systems and lay the foundation for understanding the biology of developmental diseases, cancer, and neuronal disorders. Some questions we are addressing in our lab are: how do different signaling patterns determine distinct cell shapes? How cell polarization is regulated throughout different steps of cytokinesis? And how multiple signaling proteins crosstalk to regulate cell shape and cytokinesis. Graduate students in my lab use cutting-edge live cell microscopy, genetics, quantitative protein dynamics, and more to research these processes. I hope you have enjoyed these short vignettes that sample some of the diverse research in our department. We invite prospective graduate students to apply to our graduate program. We will start reviewing applications after December 1st. Please use the link shown here to learn more and to apply. Thank you.